Yeah, so today it's my pleasure to welcome Harry Holthofer. Professor Holthofer has held visiting or professorships at Albert Einstein College of Medicine in New York, in Dublin, Ireland, and in universities of Munich, Freiburg, and Hamburg, Germany. He has set up and coordinated several European Union-funded programs focusing on diabetes and its new diagnostics. He is a founding member of the International Committee to Standardize Proteomics Methods in Kidney Research and European Kidney Health Initiative. So his current research activities has focused mostly on kidney complications of diabetes and new diagnostic utilizing extracellular vesicles. So today, he's going to give us a lecture on simplified techniques for extracellular vesicle analytics. So please welcome Harry.
Okay, welcome and good morning, everyone. Good morning from here, and obviously we are speaking mostly perhaps in Europe. So the title of my presentation today is Simplified Techniques for Extracellular Vesicle Analytics, and I want to go through some of our findings during the last years, our approach, how we feel that we have succeeded in simplifying some of the techniques, and how we think how these have to be validated. So, and as you can see from here, I'm coming from two institutes. One is the University of Helsinki in Finland, and the other one is University of Hamburg in Germany. With these audios, I don't need to go too much into detail about extracellular vesicles or secreted vesicles. Just to reiterate that yes, every single cell in our body is doing this task all the time, secreting vesicles, and really this is genetically strictly defined system how these are being done and then released to the extracellular space. And as we know, that the recipient cells, then they have various mechanisms how they take up these extracellular vesicles from the intercellular space. And as we know, they can be divided into different categories like apoptotic bodies, exosomes, microvesicles. But most important is that they have this beautiful contents of different kinds of different sets of RNA, double-stranded microRNAs, you name it, as well as proteins. And the composition of lipids is very distinct as well in these different groups. Something that we consider important is that all these contents, EV contents, they seem to be shared in all of these. So for our purposes, for mainly biomarkers, it doesn't really matter whether we focus on exosomes or microvesicles or any subsection sectors of those. It is for us, it's a less important question. And something, especially as we are working with urine, we always need to take care of the bacterial outer membrane vesicles as well, and how they kind of influence that as well. This is not new to anyone here as well. So some of the established functions and these are going to finer and finer detail, of course, is all the time. Cell-cell communication, transfer of RNA, proteolytic processing of target proteins, enablers of cancer cell spread, so metastasis, modulator of immunological responses. So coagulation and inflammation as well. So they seem to provide very valuable biomarkers, or search for biomarkers, at least at this point. And why we are interested about those is also that they can be customized to future tools for drug delivery as well. And I don't go into detail about these brilliant articles on metastasis and EV role in metastasis, or signaling in and out from the brain, as well as, and nowadays also very rapidly kind of progressing treatments where vesicles can be used, like in the Duchenne muscular dystrophy. the real narrative and ethos in my lab has been to really focus on EVs and their potential for biomarkers, as I was alluding to. Yes, methods and workflows, they need urgent attention and standardization. This is very important for us, and we have been really working hard to really fine-tune some of these workflows. But something which is extremely important for us is that the methods should be simple, they should be accurate, reliable, robust, non-invasive, and affordable. Yes, we still need agreement on what is needed for minimum requirements to ensure reliable results to be comparable to results from other laboratories. So there is lots of work to do, and of course I'm the first one to acknowledge the beautiful work that has already been done on the standardization. As I mentioned, non-invasiveness is very important for us, and the kind of natural selection for us for assays is urine. It is really underutilized diagnostic biofluid from that aspect, and we can easily understand the advantages of that. But first, couple of words about the formation of urine. It's formed in the nephrons, the nephrons in the two kidneys, and the nephrons, that there's about 500,000 nephrons per kidney, and it's everything starts from the glomerulus, kidney glomerulus, which is the filtration unit, and then the primary urine flows the proximal tubules, enters the loop of Henle, goes to distal tubules, and down the collecting duct, and finally to excretion out of the body. And of course, the obvious advantages are the easiness of collection. It's completely non-invasive. Follow-up sampling is possible, and large volumes are possible to collect as well. And this is, we think that this is very important aspect, because if we have from the very same sample, we can divide that for various techniques, various parallel and orthogonal techniques, and we can run our RNA, we can run our protein, we can run our metabolics from the very same samples, isolated the same thing. That is, in our thinking, the only way that we can use those reliably. Yes, of course, we understand that urine it has also challenges. It's considered to be a dilute metabolic waste product, and of course it is. It has broad range of physical chemical properties like pH range, which has its effects on, for instance, protein conformation. It has also, urine has also intra and inter-individual variability, which has to be considered. Environmental factors impact the composition of urine. And then something which is very important, this has been the limiting factor in utilizing urine more, is that the thumb-horsefall-glycoprotein and human serum albumin, they are the common contaminants, so to say. Then they interfere with all analytic downstream assays. And of course, the urinary pigment belongs to the same category. We have to get rid of the interference of these. This is important. Exposure to intranephron proteases, RNases, and other enzymatic activities. That's important to keep in mind. And this is really why extracellular vesicles found in the urine, they are the absolute brilliant source, because the crude urine, you get split products of proteins, you get RNA splits there and spliced RNA. So as we all know, everything seems to be so much better conserved inside the vesicles. But then again, bacteria produce their own repertoire of proteins, metabolite, lipids, as well as the outer membrane vesicles. So we have to distinguish ourselves from those as well. Okay. Coming to the vesicle isolation methods is in use, and this is from a publication a few years back, but this seems to hold true even nowadays. So golden standard is still the ultracentrifugation, serial ultracentrifugations, and then the other ones, density gradient filtration, size exclusion, chromatography, precipitation, and so on, or more and more combination of these. Depending on the availability and volume of the sample to be analyzed, there seems to be reasonable selection and basis for selection of these different methods, and you can see how, for instance, precipitation-based methods are used more in the smaller sample volumes, whereas it's much less useful in the sample volumes bigger than 100 milliliters. And we all know that the serial centrifugation, this is and will remain, I guess, the golden standard for good time to come. But again, these familiar gadgets here, they are high in initial investment costs, and they of course they also require highly trained personnel to run those samples. Otherwise, they are dangerous weapons. And something that we couple of years back with Luca Musante during my time in Ireland, we were working to really put together all these methods that were used at that time, and compare the pluses and minuses of each of these. And you can see that we considered quite a bit of these different methods in combination with the differential centrifugations. But for instance, of course they have their obvious advantages, but then disadvantages, like with the differential centrifugation, there are no fixed conditions, for instance, for the number of centrifugations used, relative centrifugation force, time for centrifugation, rotor type, sample type, temperature during centrifugation, and presence or absence of protease inhibitors. But really to summarize everything, it seems that these methods have not been applicable for large volume samples, and this is one thing. And they are not very suitable for large patient cohorts. They are relatively expensive because of the device setup and the reagent price. And to put it very simply, if this barrel here is the total urine and its contents of extracellular vesicles, we have clearly shown with Luca Musante in Dublin that the pre-analytical steps, we are losing between 20 to 30% of vesicles. The same thing is true for the post-analytical steps, 20 to 30% again lost in there, meaning that we might have 60% left from the original setup of vesicles in the urine, and we don't know what we are losing in the pre-analytical and post-analytical set. So this is a major distortion of the results. So we have to get simpler, we have to get faster to the real analytics, and this is something that we now consider that this is, at least it is simple, and we have run this back and forth and repeating and done our trials and errors, but this is the simplest form that we can think of. So as you see, and I think you can appreciate that it is very, very simple. In the Büchner bottles here, we have the urines, and this is connected to the dialysis tubing, which is inside the Erlenmeyer bottle here. And what we are utilizing is the hydrostatic force to push everything down the dialysis tube, and we may or may not add a little bit of vacuum to speed it up. It takes running half a liter of urine takes between 1 hour and 2 hours, and if we add the vacuum, it speeds it up to 20 to 30 minutes in the best case. So this is very, very simple. This is not a condom testing station, but this is rather shows how easily the HFD units can be multiplied. So here, for instance, we are running 9 patient urine samples in parallel. So it can be very easily done, and you can easily also calculate the costs of the materials and reagents and starting volumes. And note that we don't need any electricity, for instance, for this. So it has some very interesting things to follow from that. So it seems that our HFD method is quite good to optimize the EV harvest, especially from the urine. It is really simple, it's highly cost efficient, and it is fast. It can be used in very simple environments, as I told, but then we have also shown that it can be used for cell culture medium, plasma, saliva, urine, milk, and many others. So it is really versatile to use in these different settings. And of course, you can, if you have a low volume, you can dilute it and then run it through the membrane. But this is important so that this can handle large volumes between 200 and 400 milliliters or more. So it is possible to get enough of vesicles from the same single sample so that we can run different analysis from that. Of course, that increases the comparability of results, and we can work with the transcriptomics and proteomics and metabolomics and you name it from the very same sample with this setting. Of course, samples are directly concentrated up to 100 times and equalizes all the urinary solutes. It also is easy to get rid of Tam-Horsfall and other contaminated proteins, and as well as pigments, which those of us who are working on RNA know that the pigments are really the killers of the downstream assays. Yes, it can produce very useful RNA and protein biomarkers, as I will show you in a minute. Yes, and up to now we haven't found a downstream assay which wouldn't be fit for this analysis. So how do we get convinced that this is a useful technique? Well, this is how we did it. So we have our hydrostatic filtration dialysis, serial ultracentrifugations, as well as the kits, various commercially available kits. And I have to tell that we have nowadays, we have tested up to 15 of these commercially available kits for this purpose. I don't want to advertise this one. This just happened to be best in our hands, working best. And again, we are now isolating, we are using the very same sample, a little bit bigger amount of urine, and divided it to HFD, UC, and the kit analysis. And what we did was to assay for NTA, for NTA, electron microscopy, Western blot, RNA and RNA sequencing. So we think that this was a comprehensive enough set of ways how to validate the HFD and other methods to each other. So what did we find? So here is the nano NTA analysis. So you can see that the HFD and HFD and ultracentrifugations, that they work pretty much similarly, and they were able to isolate good amount of vesicles as compared to the kit-based method. With the different particle size distributions and ability to pick those, HFD and UC again, they very nicely kind of work similarly in the smaller range as well as in the higher range of vesicles, and whereas the kit-based ones they don't work that well between these 100 to 200, 200 to 300 and so on. They pretty much work the same, but it is clear to state that that really there are differences in the performance, and you have to know what you're hunting for, and based on your selection of techniques on that. We also sequence the RNA from the EVs and did the principal component analysis of that, and we show that again the ultracentrifugation with blue dots here, and is this yellowish dots here with HFD. They perform very nicely and very similarly as compared to the kit-based methods, and the same thing we can find a nice correlation with the UC centrifugation and HFD, whereas the kit-based they run differently here. A little bit different stories with the microRNA. We are screening nowadays between 500 and 5,000 microRNAs, and this is how it looks. It seems that the kit-based method it pretty much shows the same type of reactions and performance with the other methods. So our conclusion here is that really the kit-based methods in this case they seem to be performing quite well. Okay. Of course, we have this kind of validation, but then we have to bring these approaches to real life and the clinical targets. We are working in the university hospital setting, so we have access to patient samples, and this is of course important that we are working, making sense of these hobbies that we have with the extracellular vesicles. So our traditional research line has been diabetic kidney disease and diabetes and its complications, and this picture shows why this is the case. WHO did an estimation of diabetes and its diabetes burden in the world during the coming years up to 2030, and you can see that well, pretty much Europe, South America, Australia, there is moderate increase in the cases predicted, whereas especially in India, in Africa, and in China as well, the increase of diabetics kind of projected to 2030, 10 years from now, it was these numbers, so 86 million by 2030. Now, for instance, in China already, the number of diabetics is around 110 million. So heavily going over those estimations. And these 480 million global cases by next 10 years is the current used number. I'm taking the example of the diabetes in China because they are taking this this seriously. They have recognized that this is a problem. 12% of their population suffers from diabetes or pre-diabetes. 60% of cases remain undiagnosed, and this is, of course, the big threat. That leads to serious complications, and what they want to do is to avoid these uncontrolled healthcare spendings that these numbers are spelling. So there's huge need for predictive, accurate diagnostic solutions and really predictions like described here. So what we need is the predictive biomarkers before the disease is set on. Oh, sorry. And then when the disease diagnosis is made, we need to have means to monitor the clinical outcome. And then we are introducing certain medications, and we need to be able to follow who is getting any advantage of those ones. So this is the basic setting where we wanted to test the methods and especially the HFD method. So what we are doing here is that we are hunting EV biomarkers for diagnostics, for prognostics, and for new targets, new target identification for new medications. And how we are doing that, we are using the in vitro model. So we are culturing the diabetes prime target cells, and then we are harvesting the vesicles out of them. And I need to add that we have the cells and cell environment. We can engineer that to resemble insulin resistance and insulin sensitivity, and we can transfect those with insulin receptor, as I will show you later. Something which is important for our thinking is that, yes, we want to see if the in vitro models that are being increasingly used for diabetes targets and milking out the extracellular vesicles for that purpose, if that can be confirmed in the established in vivo models, like the streptozotocin diabetes model in the rat, and especially in the urine of the rat. So seeing that there is these hold true, these two cases, and then also that having the information from these sources, how we can kind of translate that in the clinical sample analytics. The final goal is to see whether we can simplify and really cut corners in the and replace the collection of heavy clinical sampling to really model the disease effect in cell culture, and that seems to be possible, as I will be showing you. What we are doing there is the messenger RNA sequencing. We are doing microRNA, and we are doing quantitative proteomics in the same setting. So and these are all from the extracellular vesicles in these three different settings. And again, this is what we are targeting. It's known that diabetes hits the kidney glomerulus, the filtration system there, and especially the epithelial cells, which are called podocytes, the endothelial cells, which align the vasculature in the glomeruli, as well as the mesangial cells, which are really the chemical factories and support cells, which are keeping this system together in the single nephrons. So what did we find in the DKD, diabetic kidney disease target cells, podocytes, mesangial cells, and our control cells, proximal tubular cells? We confirmed that, yes, indeed, we have vesicles. We have vesicles typical of exosomes and larger as well. And then we showed that TSG101 in podocytes, mesangials, proximals, as well as endothelial cells. And as you see, in all of these settings, whether it's insulin sensitive or insulin resistant cell environment that we are culturing them, or we have transfected with the insulin receptor into these cells, we do have the vesicles coming out. Not too much difference in the amount of vesicles secreted of each of these. In NTA, yes, we show that really in these podocytes, insulin resistant cells with insulin kind of transfection, insulin receptor transfection, that leads to increase of vesicle secretion, and that is true for mesangial cells and proximal tubular control cells, but not the glomerular endothelial cells. So this already kind of shows us that, yes, there are differences in the cell types, how they are affected by diabetes, as the readout is the vesicles, and this turns out to be very practical information to go further. From the microRNA assays, at that point we had the 96 microRNA assays. We can see that they are nicely distinguishing in different sets of microRNAs, each of the cell types. Red is podocytes, this color is mesangial cells, and proximal tubules blue. And you can see that with the principal component analysis, driven analysis, we can see that, yes, these cell types, they really can be separated from each other based on the microRNA contents. If we go further into detail about the insulin sensitivity and insulin resistance in each of the cell types, you can see that they are more mixed, and that is what we actually expected. But you can also see that there is a trend that also these different conditions like insulin sensitivity, insulin resistance can be described by these different sets of micro RNA markers in these different cell types. These don't necessarily overlap between cell types, which is also interesting. Yes, we are doing quite a bit of proteomics at the same time, and you can see that we are doing with the different settings in different cell types. We are doing here like with the proteomics here of podocytes, and we are doing with the basal conditions, insulin resistance, and the other sets, and we can really identify the different markers in these different settings, which means that we can then develop these lists of proteins which are increased or decreased in these settings. And of course, we are interested about the protein networks. This is what we can easily develop from these, and then of course important is that our microRNA results they really keep together with the protein data, which means that we can identify microRNAs which are leading us to certain key molecules and molecular networks that we are working with and identified in diabetes in this case. So this seems to be very valuable way to really go from the simple setting of diabetes target cells, milking their vesicles and analyzing those, instead of going always to the heavy patient studies. Yes, indeed, we can identify the cells, the pathways which are involved in these different cases, in different cell types, and this is something which is not only cool information, but it's something which really seems to add new dimensions to our understanding of this whole disease. Yes, indeed. The extracellular vesicles in the culture medium, they reflect the cell type specific reactions, also the diabetes conditions in each of these, and they absolutely, they seem to be a nice robust shortcut to diabetes research. And the same thing, so this was about the cell culture studies, and as I told earlier, we consider it important that the results here are in keeping with the results from the in vitro models. And this is, I'm not going deeper into these ones, either the experimental models on the human results that we are increasingly generating now. Just to point you out that this is really something which seems to work. There is good agreement with the results and pathways identified between all these settings, and that means that target cells are becoming more and more important in the future for analysis. This is just going deeper in the rat models, rat urine models, and what we can find out of there, and so that we can really confirm results in these different settings. This actually shows our kind of mind map. So we are starting with the diabetes and diabetic target organs. Like here it is the kidney. Yes, it is indeed a global problem, increasing problem. If we want to really combat this problem globally, we have to be simple. We have to be able to monitor these changes very easily, completely non-invasively, and the URI seems to provide that solution. Yes, indeed, we have to do our homework carefully in the lab to identify reliably the markers, the biomarkers which are associated with these complications and others, and then we have to define the way how we want to analyze them. In this setting, we had been testing with the microflow chamber systems. We are adding the urines, the EV containing samples here, and we are checking the biomarker, distinct biomarkers in these different wells here. And of course, the key idea is that there is an easy readout in the future to our mobile phones, for instance, wirelessly getting the results to mobile phones. And then, of course, if there's something wrong in your diabetes status today, or some other chronic disease, the critical information is delivered to the healthcare system. To keep up the idea of simplicity, this is showing our approach, how we are an alternative, how we are doing the biomarker measurements. So that there are spots in these paper-based printed channels where we can distinguish, we can measure our favorite biomarkers in these spots here, and you can guess that this is printed paper. The size is like here 2 by 2 centimeters, and it's very easy. It's basically filter paper which absorbs automatically the urine and vesicle sample, and then it delivers it to these different. The cost is negligible. It's zero cost with this approach. So in conclusion, simple techniques and workflows are available for EV isolation and the workflows for essential downstream applications. The techniques, however, they always need stringent validation and standardization. And orthogonal techniques, including proteomics, agronomics, needed for reliability of results in each of these studies. And something which we find very important, that we also, especially when we are using the patient samples, we are including both normal and patient samples, patient urines in these situations, because they behave quite differently. The urinary environment as well as the serum environment is different in normals and in patients. And of course, what we are trying to find is how can we normalize the situation so that we can really pinpoint the distinct biomarker from there. And of course, urine appears to be very well suitable to hunt for these disease biomarkers, and this is really we think that this should be the preferred choice for mass screenings. And there are techniques, as I showed you, the HFD as a prime candidate to do it very simple, very cost efficiently. And finally, my acknowledgements to my group in my years in Ireland, in Dublin, particularly Luca Musante. In Bristol, they have been engineering the diabetes target cells. Very good collaboration with them. And in Hamburg, my present second working place, the group of Tobias Huber, where we are working very heavily with the single cell analytics and imaging. And in Helsinki, especially Karina Barreiro is the powerhouse, and our funding comes mostly from European Union and the Novo Nordisk Foundation. And with that, I thank for your attention.
Thank you very much, Harry. So this presentation is open for discussion and questions. And I ask everyone to please post your questions in the chat, and I will allow you to unmute and ask your question in person. And I begin with Rosella Crescitelli from my own lab.
Yeah, very clear presentation. Congratulations. I followed almost everything.
Thank you, Rosella.
I have actually two questions connected to the technique to isolate vesicles. Because you say that your HFD method can be useful to isolate vesicles from plasma, serum, saliva, and so on. So the first question is, you can save your life because we spend most of our time to isolate vesicles. So if it works, it's great. And do you have results about vesicles isolated from other source, not only from urine? And the second is...
Take the second question later.
Okay.
Go ahead, Harry.
Yeah. Yes, thanks, Rosella. They really important questions. We do have results from saliva, serum, and cell culture medium and urine at this point. We are working a little bit with milk as well.
And they are
so the big issue we have is that the lipoprotein particles co-isolate with the vesicles, and that's why we're pushing so hard to separate them. We want to understand the details of but maybe it doesn't matter if you're looking for a biomarker. Is that what you're saying?
Well, this is exactly so. So we are really not so we just are hunting for the biomarkers. That's true. So it's kind of a bulk issue. We are just working on those. But just to in a way comfort you that this method, our method, it's very easily modifiable. We have been forced to really work with Tam-Horsfall protein and some of the other contaminants which we have managed to get rid of with a little bit treatment here and there. So I guess that could be modified to a variety of targets.
Your second question, Rosella?
Yeah, you show that size of vesicles measured by NTA were different when the vesicles were isolated by your system and by ultracentrifugation, and they were bigger when isolated with kits, if I understood well. This is a big surprise for me because I never seen different in size in vesicles after measurement with NTA. So do you have an explanation for this?
Well,
why they are so bigger, or why the kits are able to isolate vesicles that are so bigger than?
Well, that's again a good question. It seems that the HFD and ultracentrifugation, we are able to really harvest the whole repertoire that there is, whereas the kits they tend to be, I don't know, deliberately or something else. Of course, their capacity is also small, but they seem to be performing better on certain size vesicles. So that is my only answer at this point. Sorry.
Yeah, no, but my question was not really connected to the way to isolate, because it's clear that different way has different. They are more good. They are better to isolate vesicles bigger or smaller, but was connected to NTA, because we never seen a different in size, also loading different kind of samples that we know that they are different size. Maybe it's not for you the question, but for your guys working in your lab. But
yeah,
you know how they manage to have this different in size,
yeah,
because we've never been able to see this such different.
Right. Yes, I get this is a question that we have to think in the lab as well. But this is really the results what we are getting. How to explain it? Hard to explain. Yeah.
Okay.
So the bias we have, or the experience we have, is that NTA is reasonably good to count particles, but not reasonably good at looking at size. So when for example, Rosella or Cecilia look at electron microscopy pictures and measure the diameter, we see differences between different isolation methods. but the NTA doesn't show any difference. Then again, there's this overlapping Rosella, so it's yeah. So that's our experience with NTA. I mean, it is useful for counting, but not for size. That's what our prejudice anyway.
Yeah, well, this pretty much holds true with our ideas as well. So to be honest, we rather count on the electron microscopy what is the scenery in there, and then combine that together with NTA. Yeah.
Okay, I think we can move on. This is a tough question. It comes back every week almost, so I'm sure we will be back on it. But Maya has a question, so please unmute yourself and ask a question.
Hello, everyone. Harry, that was a beautiful lecture and very interesting.
Hello,
so I have a question about the technique, actually. What was the cutoff of your dialysis tube? And whether you've done any recovery experiments? What is the recovery?
Right. Maya, thanks for this question. And please believe me, we have really been working with the different cutoff membranes, and with some reasoning and some unreasonable results from there. But the cutoff that we are mostly using is 1 million kilodaltons, 1 million daltons, sorry. So that is the one that we are routinely using. For special purposes we are doing other cutoffs with the membranes as well. But yes, this is the mostly used one. Yeah. Sorry about your second question.
Have you done any spiking experiments? What is your recovery?
We have those, and now I should be connected to someone in the lab, but I'm not. So I could, if you can share me with your email address, I will be giving you the numbers of that.
Okay. Okay. Thank you.
Thanks.
So
Maya.
I have another question, which is relating to urine and biomarker discovery there for other diseases than kidney-associated diseases. I'm a clinical doctor background, so my prejudice is that very little material from the circulation leaks out into the urine. And you were also saying that most of your vesicles are actually coming from the kidney. Is there any possibility we can find biomarkers for non-kidney associated diseases in patients that don't have a severe glomerular disease or something? Because otherwise,
absolutely,
it would be stuff, right?
Yes, absolutely. And as you know, as a earlier clinical doctor, this is a wonderful window. Kidney is a wonderful window to cardiovascular diseases. So whatever concerns cardiovascular diseases, you get them. You get very surprising molecules in the urine in the different cardiovascular diseases. You get leakage of those which are supposed to be from the heart, like the BMPs. You get leakage of those to your urine in the vesicles, not in the crude urine, but in the vesicles. In liver diseases, which is very common nowadays, as you know, it's one of the hot topics in clinical medicine. Yes, we find liver specific ones in the urine as well. Then something which we are increasingly working on is the pulmonary diseases for certain reasons, and brain diseases as well. So we want to replicate certain settings in the brain diseases, and we have focus on those. But in addition to this, we are working with diseases like chronic diseases, like autoimmune diseases. Celiac disease is one of those. We get nice positive results. So to answer your question, yes, indeed, you don't need to have a kidney disease. it looks like there is always kind of passage of vesicles from the circulation, whatever the mechanisms are, how they are selected to be kept in the circulation, and what are the ones which are released to the urine, which are then processed back to the proximal tubular cells or the others. We don't know about those, but indeed there seems to be many ways how the vesicles from circulation get access to urine, and they are protected there until they avoid it out. Why
so they're not leaking? They're not leaking out into the urine. They're actually actively transported to the urine.
As well. There seems to be transcellular transport in the proximal tubular area. They are taking advantage of the built-in cell biology mechanisms there, from the base of the cells to the apex of the cells, and to be kicked out, spat out there. But the kidney glomerular filtration barrier, it seems to be selective for some of these. How, why, and how is it working? We don't know, but this seems to be the way.
Any more questions for Harry? There's one from Carolina Soekmadji. Please unmute yourself.
Thank you so much, Harry. It's such a wonderful talk. I think I'm just very interested with this urine source of EV. So it is a potentially a very good source of biomarker, but urine is being secreted as a waste from the body. So I was just wondering whether they have some sort of biological functions, or is there anything that they can do?
Thanks, Caroline. Wonderful question. Wonderful question. Our brains are exploding with those ideas now. How that there seems to be a role for those. They are not there just for fun. Why would nature and our body waste valuables to be secreted out, unless there is a way how to utilize them? There seems to be a way how the distal tubulus is harvesting vesicles back, whether it's the microRNAs to modify the local events, or are they secreted back to the circulation? We don't know yet, but this seems to be one of the ways. How they are picked up by the collecting duct cells, we don't know. Is it electrostatic adherence? Are they more actively taking up there? We don't know that either. But definitely there seems to be always that kind of backwards flow of vesicles. They are not all wasted to the urine, but then in the urine, it's fascinating area as well, because in the urinary bladder, it's of course our normal barrier to outside world. There is always bacteria in the urinary bladder, and our vesicles need to feed the bacteria and in a way fight against those vesicles which are actively secreted from those resident bacteria in the urinary bladder. How they are doing that, we don't know. But of course, the big question there is that when these bacteria in the urinary bladder are winning the game, to our vesicles secreted all the time, constantly to urinary bladder, then it means that there are infections. We are getting cystitis. We are getting ascending urinary tract infections. So it's a fascinating area, and we believe that there is always these kind of feedback systems, loops, whatever they are active from the point of view of the vesicles secreted either actively through the cells in the proximal tubules, or passively through the filtration barrier. But then they meet their challenge in the urinary bladder. They have to fight the bacteria. They have to keep the balance. But we don't know details about that yet.
Wow, that's very amazing, fascinating.
We are fascinated that, yes.
Alrighty, thank you so much. Before I give the word to Carolina back, I would like to say to you, Harry, thank you. And also Maya's email is in the chat.
Okay.
And so you can contact her. And then Carolina, please.
Yeah, thanks so much, Jan, for leading the questions and answer. And Harry, again, thanks so much for opening our minds for what urine EV might do, and also as a good source of biomarker and what it can do to isolate them. Thank you so much for that wonderful talk.